Hi everyone, I'm Dongmei Zhi from Beijing Normal University. I'm currently working with Professor Jin Sui. Now I'm going to talk about my project using resting-state functional connectivity to identify depression subtypes with higher ECT response rates. In this talk, I will briefly talk about the background and the problems, then describe the method adopted to solve the problems. Finally, I will demonstrate some key results and conclude my talk. As is well known, depression is characterized by persistent anhedonia and emotion deficits. Depression is a largely heterogeneous mental disorder driven by varying clinical symptoms. And depression is often comorbid with other mental disorders, such as generalized anxiety disorder. Most importantly, depressive patients often respond differently to a given specific treatment or intervention. The large heterogeneity of depression seriously hinders precise diagnosis of depression, which may delay timely and optimal treatment. Moreover, the number of depressive patients is increasing year over year, causing severe social and economic burden. So our challenges are how to objectively pass the heterogeneity of depression and seek the optimal treatment for each subject. Recent studies have tried to find the best therapeutic targets and pass the heterogeneity of depression using brain neural imaging. For example, functional connectivity between subcallosal cingulate cortex and the above three regions were associated with remission to antidepressant. Specifically, the remission rates of an individualized treatment can achieve at 86.4%, targeting at the regions of left dorsolateral prefrontal cortex, most anti-correlated with subgenual anterior cingulate cortex for each participant. Additionally, 4 subtypes of depression were identified by combining functional connectivity and depressive symptoms, and showed different response to rTMS treatment. And 6 brain circuits were defined for depression and anxiety, and can distinguish response to antidepressant and behavioral intervention treatments. Taken together, the data imply that functional connectivity can partly pass the heterogeneity of depression, showing greater potential application in individualized treatment. In this study, we aim to use functional connectivity to pass the heterogeneity of depression and identify replicable and generalizable depressive subtypes. Further test whether distinct subtypes have different behavioral manifestations. Most importantly, we explored the specificity of each subtype in response to ECT treatment. First, whole brain functional connectivity was estimated for each subject based on AAL atlas. Then, we used non-native matrix factorization to divide all participants into subgroups in the discovery and replication datasets respectively. Non-native matrix factorization is a statistical approach that decomposes data into non-native parts and allows us to simultaneously cluster features and individuals into subgroups. A particular advantage of path-based representation of NMF is that it can mine similar aberrant patterns within only a subset of groups, and such nonlinear relationship cannot be detected based on global similarities. In 2 independent datasets, 3 clusters consistently showed an optimal fit of the data with high stability, which were dominated by frontal-parietal, contralateral, subcortical occipital connectomes, and a high spatial correspondence was observed for the corresponding subtypes, with dice similarity coefficients of 0.81, 0.81, and 0.78 respectively. Similar results were observed at node and network levels. Subtype 1 were represented by connectomes among default mode, lateral attention, frontal-parietal, and sensory motor networks. Subtype 2 were represented by connectomes within contralateral networks, and subtype 3 were represented by connectomes between subcortical and visual networks. We further conducted a neuroscience meta-analysis to seek the representative behavioral topics associated with connectomes of each subtype, suggesting that each subtype was linked to different behaviors, namely listening perception, face recognition, and episodic memory, and reward anticipation respectively. Additionally, a linear support factor MAC was used to test the discrimination of the three subtypes within and across datasets. Results showed that SVM yield high accuracy of 89% and 88% when classifying three subtypes within each dataset, and 86% and 83% for the across dataset prediction, suggesting high replicability of the derived subtypes from adaptive data class 3E. Compared with Hessian controls, subtype 1 showed hyperconnectivity between default mode and ventral attention networks, between default mode and sensory motor networks, and between frontal parietal and sensory motor networks, especially the prefrontal parietal connections. And subtype 2 showed prominently decreased connections between frontal parietal and visual networks or sensory motor networks, and between subcortical and visual networks, especially the striatum occipital prefrontal hyperconnectivity. For subtype 3, increased connections were observed between subcortical and visual networks, while decreased connections between limbic and sensory motor networks, and between visual and sensory motor networks. After obtaining the cluster-defining features for each dataset, the cluster signatures for each subtype were finally denoted as average connections of the overlapped cluster signatures across datasets. Most importantly, to identify the most treatment-responsive depressive subtype, we mapped unseen individuals receive ECT treatment into their corresponding clusters using the cosine distance of cluster signatures to the closest centroid. Results demonstrated that the response rate of the subtype 2 was higher than the other two subtypes with ECT treatment, while subtype 1 showed the lowest response rates, implicating that depressive patients classified as subtype 2 could be more likely to recover after ECT treatment. And here is the conclusions. We identified three reproducible and generalized subtypes for depression across datasets, which were represented by frontal parietal, contralateral, and subcortical occipital connectomes respectively. Different subtypes were associated with distinct behaviors, namely listening perception, face recognition, and episodic memory, and reward anticipation respectively. Especially, the frontal parietal connectome subtype had the lowest response rate, while the subcortical occipital subtype had the highest response rate to ECT treatment. Finally, for this project, I would like to thank my advisor Jinsui and all collaborators. Thank you for listening. If you have any questions, please feel free to email me.